Streptobacillus rodentia is a novel gram-negative bacteria recently discovered to have zoonotic capabilities. Characterized by the phylogenetic order from which it was discovered, S. rodentia was recently cultured from the oropharynx of a community of capybara and several beavers within the Roger Williams Park Zoo in Providence, Rhode Island. A genetic variant of Streptobacillus monoformis, S. rodentia is harmful to its host and displays similar symptoms of Haverhill fever. It's a novel strain with a newfound ability to frequently infect larger rodents and pass them from animal to human transmission. Common symptoms include fevers, rigor, headache, rash, and polyarthralgia. In addition, researchers are also identifying respiratory complications similar to tuberculosis and pharyngitis. S. rodentia may also be treated with antibiotics such as amoxicillin, penicillin, and others. Alcohol-based antiseptics and disinfectants might prove more successful by targeting the lipid bilayer of the cell membrane. S. rodentia is a species of the genus Streptobacillus, indicating a chain of rod-shaped bacteria that form into loosely tangled multicellular clumps. Their average size varies from 2 to 5 micrometers, with long curved segments or groupings of 100 to 15 micrometers. Each individual bacilla is straight, but is capable of developing lateral bulbar swellings. It's also capable of inducing endotoxic shock if it's lysed. Research have also discovered that the presence of unique lipids containing mycolic acid, which has pathogenetic capabilities within it. The presence of fibrillae enables it to adhere to epithelial cells and components of connective tissues, particularly among the epithelial tissue of the respiratory tract. In addition to the boundary layers, the glycocalyx surfacing coatings functions as a barrier between the internal and external environment and serves for the formation of biofilms. And then there are inclusion bodies and microcompartments that carry out storage, and S. rodentia is unable to undergo sporulation. The genetic makeup and structure of S. rodentia involves a singular circular chromosome located in the nucleoid region. The 16S rRNA gene was sequenced to identify it at the species level. Many clinical laboratories rely on this method to identify unknown pathogenic strains more specifically. The following lists here you see are all the genetic characteristics of the chromosome, but I won't go into them in detail for time's sake. Production of the functional proteins occurs via transcriptional translation in the cytoplasm. Introns and splicings are not transpired during this process. There are several inducible operons activated by the presence of enzymes found in the mammalian respiratory tract. It's also able to produce secretion proteins that bypass the inflammatory responses of the host cell. S. rodentia undergoes semi-conservative replication, but has a unique capability of initiating at 2 origins of replication, which speeds up the growth process. S. rodentia is a microaerophile and does not grow in atmospheric concentrations of oxygen. It is also a capnophile, which means that increased carbon dioxide is preferred. The ideal temperature that it grows at is 35 to 40 degrees Celsius. Its ability to establish intracellular bacterial life cycle depends on several mechanisms, including invasins that permeate the respiratory cells. Because S. rodentia is unable to synthesize organic compounds as a heterotrophic organism, precursor molecules will undergo anabolism to establish the building blocks for future cell replication, including amino acids and nucleotides that await assembly into macromolecules and other essential components of the daughter cell. Amphibialism can be seen amongst cultured S. rodentia and is a key factor for efficient mechanisms towards replication and survival within the host. S. rodentia resides in the oropharynx of select group of rodents. It's only known in the Streptobacillus configurations. It's likely that healthy rodents serve as the primary reservoir and will demonstrate the organism in cultures of the nasopharynx, larynx, upper trachea, and middle ear. The combination of carbon dioxide concentration and the presence of glucose within the airway surface liquid proves to be an ideal ecological niche for S. rodentia. It is this complex set of environmental conditions that kind of explains the pharyngitis and the complications with tuberculosis and other related symptoms. Relating to the interactions with the immune system, roaming phagocytes will use the MHC2 complex to identify antigens derived from the bacteria. However, the intracellular nature of the bacterial does not expose itself in appreciable amounts. So those living inside the cells may interfere with the expression of antigen-presenting molecules, inhibiting the T cell activation. The bacteria is also able to hide from MHC1 processing pathways within tight phagosomes, but despite these hindrances, all intracellular bacteria induce CD4 and CD8 T cells to assist in binding. Non-infected cells will engulf the bacterial antigens and undergo apoptosis induced by intracellular bacteria. Another form of immune evasion from S. rodentia comes from the production of effector proteins that are secreted to inhibit immune response. Once the immune response has been established, an inflammatory response kicks in, and it will immobilize and attract immune components, including basophils and neutrophils. Macrophages serve to engulf and destroy pathogens and debris left by inflammation. A fever will also be induced by the release of exogenous pyrogens from the lipopolysaccharides of the bacteria. They'll inhibit multiplication and availability of the iron, making replication difficult for the bacteria. And then antimicrobial peptides, including defensins and protegrins, are especially effective against this bacteria. They're short proteins capable of inserting themselves in the membranes and disrupting the membrane and compromising that membrane. These are commonly found in the phagolysosomes of the host.